was one that was incubated at 44.5 degrees. I probably said 44 degrees when I told you that. Okay, because organisms that have been in the intestines are more likely to be tolerant of a higher temperature than, say, coliform-like organisms that came from insects or from debris of some other type. So it's a subset of coliforms. And I think I brought it up because I was saying that, I don't know, there was some like number that I was reading you of the number of fecal coliforms in something. Okay, because if they're fecal coliforms, like I don't know if I mentioned this one, but in Oregon they have for a hamburger, for ground raw hamburger standard. Most places don't have one, and it was 1 million fecal coliforms per gram. So then it kind of gives you everybody all of a sudden is all sugar. So why don't you get sick when you eat hamburger? Because you cook it, right? So it doesn't matter that they're all there, but fecal coliforms just mean that if there's that many fecal coliforms, there's a lot more what they would just refer to as total coliforms. Total coliforms. Okay, fecal coliforms a subset. So fecal coliforms are also used as an indicator too. I think that's all that I can mention. This is just one of the other tests that are being used. What's required though is the most probable number. That's the required test. All the other ones are ones that the water treatment facility would prefer to do to try to help, because the water treatment plant, they don't want people getting sick. They don't want to issue a boil order. A boil order means when a public service announcement comes out telling everybody to boil their water. This happened when we had the earthquake. This has happened in other areas when they've had like a break in a water main or something like that, and you'll get an alert telling you, boil your water. Okay. Does boiling your water sterilize your water? No, but it will kill the pathogens. Okay. If you ingest a spore-forming organism, it's not going to be a problem. If it gets into something that's going to be kept anaerobic, that would be a problem. Okay, so any questions about water? So also, in reading your results for water in lab, since we already mentioned about the most probable number, for your membrane filtration, it's going to be the number of colony-forming units per the amount of water you filter. So when you write it up on the board, you know how to write up the membrane filtration, and you know how to write up the most probable number. Okay, so membrane filtration, the number of coliforms, colony-forming units of coliforms, per the number of mLs you filter. If you didn't pay attention to that, you could just estimate that. Like if you filtered around 50 mLs or something, it would be based on that. Okay. If you don't have coliform-like colonies, then that wouldn't have anything to read from there. Okay, so any questions about that part? And then for your last one, you just put 1 ml of water, and you were looking, that was the heterotrophic plant count. That is just going to be the normal colony forming units per ml. What happens if you have too numerous to count on your water sample? Should you just write too numerous to count? Or should you write something like greater than 300 colony forming units per ml? Good news. Greater than 300. Greater than 300 tells you, you know you have more than 300, because that's the only way that it's too numerous to count, right? You have to count between 30 and 300. If it's too numerous to count, it has greater than 300. That doesn't mean much when you're measuring 1 ml, but if you were doing a dilution, like in milk, say that you had too numerous to count on all of your plates. You would choose the plate that had, that was the most diluted, right? In a regular good milk sample, that was 1 to 1,000. 1 to 1,000. So if you had too numerous to count on every single one of them, and you just said too numerous to count, then you wouldn't have anything to go on. Is that too numerous to count at 1 ml? Or is that too numerous to count at 1 10,000? Or 1 1,000? 1 1,000. So if you multiply 300 by 1,000, how much is that? 300,000? So saying that you have greater than 300,000 per ml now all of a sudden tells you a pretty significant piece of information. Do you understand what I'm talking about? You kind of want to know, you kind of want to have some idea. And whether it's greater than 300 or greater than 300,000, I think there's a huge difference between those two numbers. So when it's water at 1 ml, saying too numerous to count, you know, it's not that harmful to understanding if you knew that it was 1 ml that you plated out, but saying that something has greater than 300,000 per ml, now all of a sudden you have something important that's maybe not very accurate, but it's a lot more accurate than saying greater than 300. So therefore, now you know how to report the 3 water samples, and you know how to report the milk samples. So when you go to write them up on the board, because we went through them kind of detailed, then you'll know how to do that. Because normally what happens is we keep looking at the board and then it's like, what does that mean? What is that on there? And everybody's confused. So I want you to make sure that you write your results in a way that is the proper way, and that it's going to be something that makes sense to everybody. So you're going to have to put, what is your sample? Like, was your sample, you know, out of date milk, goat milk, sitting in a car for 4 hours or whatever? A little description and the number of colony forming units per ml. If everything was too numerous to count, greater than the one that's at the most diluted, because that obviously makes the most sense. Okay. The water, there's 3 different things. So I don't think you didn't do anything different for your water samples. Like, you didn't use like one sample for membrane filtration and another sample for something else, right? You use the same sample. So we'll put the water sample, then we'll have a most probable number, a membrane filtration, and a heterotrophic plane. And you understand why you would do the 2 for the coliforms, right? One is a required test. One is a test, you know, to try to get quicker results. One is a test that just tells us the general quality of the number of heterotrophic organisms that are in there. It's nice that it's named a heterotrophic plane count, because that kind of tells you something about it. Remember when we talked about it in lab, it wasn't, you know, the low nutrient organisms, it's not psychrophiles, it's not other kinds of organisms, because obviously there are other types of organisms in there that you would expect to find. Okay, so heterotrophs, that tells us it's not the photosynthetic organisms, it tells us a lot. Right? Or we could call them chemo-organo-heterotrophs. And you would remember what that meant. Okay, so any questions about that part? Okay, so I know it just makes more sense for me to be able to mention that about the samples in here just while we're talking about that, while I have undivided attention and I'm not trying to compete with the air vent in the lab. So you guys don't have any questions about that, right? So you're gonna be looking at all your samples, counting them, sharing them with each other. I would set things out in order. Make sure you take a look at them. This is like fun, and we did a lot of extra things in here too. We had an opportunity to do some extra foods. It'll be interesting to see what the pizza looked like in here, because I know they did pizza in the other lab, and they also did spinach in the other lab too, so I'm curious to see the spinach results. Because in one of the other labs, their spinach came out, it was worse than the raw ground beef, so we'll see. I mean, I've definitely seen that before. Worse in the fact that there was more of it. But talking about like disease causing and the immune system hopefully then takes all the concerns you have about hearing these things and helps you really understand that just because there's a lot of organisms doesn't mean they're pathogenic organisms. Doesn't mean they're harmful, doesn't mean that you're going to get sick from them. However, the more organisms you have, wouldn't that maybe statistically probably increase the chance that maybe something could be a pathogen? But then again, yeah, something could be, but there needs to be an effective dose still. I hope in all of the information that I'm trying to get through to you guys, I hope too that you're also getting that ability to evaluate and assess things and come up with a reasonable way to interpret all this information. Because it isn't really just to scare people or make people concerned about things, but it's to help you like also have the ability to assess that and decide when you should be concerned about something going on. Remember I said one time that I gave like sometimes statements and asked you to respond to those statements? On some finals I've said like, okay, respond to this. What do you do, with the 5-second rule? How do you interpret that? You know what the 5-second rule is, right? You drop something, do you pick it up and still eat it in 12 seconds? After having this class, you have a lot of facts in your mind that could help you assess that. And do you think that there's a right and wrong answer to that? How many people think that there's a right answer? Yeah, there's no right answer to it, right? Doesn't it depend on a lot of things? Primarily like where you are, a bathroom, a kitchen. What do you think? Would you like drop something in a kitchen like at a restaurant and still pick it up and eat it? No. What about your living room? Maybe I'd be a lot more likely in the living room than a kitchen in a restaurant. Why not a kitchen in a restaurant? You're thinking of like all kinds of raw things that could have been there, raw meats, dripping of things, lots of organic material, but like really on your like living room floor, but that's carpeting, we have dogs. But still, it's not the same thing, right? Are there a lot of nutrients there? Is it a good environment, like the right temperature? Is there like a lot of organic material that's going to really provide moisture for organisms to grow? What kind of food is it? And that kind of leads us into this chapter. What about food, like we talked about in lab? What about all those foods would make something seem more likely to grow organisms than not? So in this chapter, they bring up a lot about spoilage too. Okay? And there's a big difference between food spoiling and food having pathogens in it. So sometimes like it feels like that kind of overlaps a little bit, but if you look at this, when you download this for food, I want you to know the intrinsic and extrinsic factors in spoilage. Intrinsic factors are going to be the things like what it is about that food, like the moisture content. Does it have a skin on it? Is it cooked? The extrinsic factors would be other things that you would, like the humidity and temperature or something. Then name a few antimicrobial molecules produced by foods. There's going to be a list on here. Understand the difference between food spoilage, bacteria, and pathogens. Has anybody ever eaten spoiled food before? You pulled something out of your refrigerator and you took a bite out of it and you're like, oh, that's gross. Yeah. Did you get sick? Yeah, it's food spoilage bacteria. They're not the same things as pathogens. Maybe you felt like getting sick because it tasted so gross, but food spoilage bacteria don't normally make you sick. It's not the same thing as a pathogen. Know a few methods of controlling bacterial growth in foods. Know a few foods or bacteria used in the production of fermented foods. And then about foodborne illnesses. The difference between foodborne intoxications and foodborne infections. So that's the chapter on food. So when you look at these intrinsic and extrinsic factors, the factors related to the food itself, and the extrinsic are the things that we would control, like the environment. So intrinsic would be things that you would really look at, the food composition. And this, again, gives information about also types of food spoilage, which I know I went through that quickly, but I'm not going to ask you to know everything about food spoilage. The thing that you have to know in better detail is understanding the difference between foodborne infection and foodborne intoxications. But here, it's here, so you can take a look at it. But intrinsic factors and having an understanding that, yes, they do degrade differently just because of those types of molecules, carbohydrates and proteins and such. So what the food is made up of, that's going to make it more likely to break down than something, that's made up of something different. So if you have something that's really dry without a lot of water content, it's not going to spoil as quickly. And when it does spoil, it's a different type of spoilage, more likely to get things like molds. The type of pH, if you have something like with a really low or really high pH, that's going to reduce the ability of certain microorganisms to break it down. If it has a lot of water, it's going to be more subject to degradation than something that's really dry. The oxidation reduction potential, if I can think back into like 107, if you have the food that's cooked, it's going to be more readily available for organisms to break it down. The physical structure, this is really important. And this is like come up, when we talked about ground meat, which is, such a big concern. Ground meat is one of those foods that is going to be the most likely to be contaminated. It's a great source of nutrition. It comes from an animal, so it's already naturally going to be more likely to be contaminated. And it's been so handled. Every single piece of it, it has touched some kind of blade. You've increased the surface area on it, so then now you can have organisms growing in all of the surface. That makes it really different than, say, a roast or a steak, where the contamination is going to be on the outside. You're not going to have all the surface area for things to grow. If something has an outer skin, so I mentioned like, don't bring a banana or something, because banana, unless we want to see what's on the outside, there's not going to be a lot of organisms growing on the inside. And again, if you're traveling, you look for things that are going to be protected because they do have a skin on the outside. I mentioned when I read through the study guide, antimicrobial substances, so it said name a few antimicrobial molecules. This is because there are a lot of naturally occurring compounds in certain fruits and vegetables and things like milk and eggs and stuff that make them at least have some antimicrobial properties. Coumarin, lysozyme. Lysozyme, we know, breaks down bacterial cell walls. That's in egg white, that's in cow's milk, that's in saliva. It's It's also really in high levels in breast milk as well. Aldehydic and phenolic compounds in different herbs and spices, this allicin and garlic, polyphenols in teas. This could go on and on and on. This is one page that's out of your textbook. But for any of you who are interested in nutrition and looking at, you know, more natural remedies for things, you can read about a lot of foods that have a lot of antimicrobial properties. So this is really just kind of a few examples that have been proven scientifically to have these properties. We do know that just historically cultures have used a lot of spicy foods, garlic, onions, and different types of chilies and everything that probably have just been known for centuries to help preserve the food or prevent it from going bad. If you look at a lot of areas that have like super spicy foods, that they just thought that that has been something that that culture developed to help prevent, you know, so many infections in foods. I mean, from foods. Again, the extrinsic factors, how it's being stored, if there's a lot of humidity, the atmosphere itself, which kind of leads us to the idea of being able to use that to help prevent flu as well. In controlling food spoilage, let's see, what's our I mean, that would normally be things like looking at pasteurization and other processes, using refrigeration, cooking things or keeping them at a higher temperature. We talked also about, you know, canning foods and how it would have to go to such a high heat to kill all the spores. Dehydrating, adding chemicals to food to prevent that. So what's on here is know a few methods of controlling bacterial growth in food. So if I asked you a question, you would just mention any of those few things. So all these past slides, using chemicals, using dehydration, using types of packaging and such. Using radiation. I did try to look up some additional information on radiation. I got all kinds of like conflicting information. Basically though, what I thought, which was strange because I couldn't find it again, I know that a couple summers ago, there was something on the news saying they no longer had to put that radiation sign on foods that have been irradiated, which what they don't have to do is they don't have to put it in unless the whole food has been irradiated under so much sugar. Anyway, it looks like a little flower. It has a circle and it has two little leaves and it has a major circle around it with lines in it. Have you ever seen that at a grocery store or anything like that? Something that says it's irradiated? Everything is been approved by the FDA for a number of years to irradiate food. Spices are regularly irradiated, and I've never seen it on a spice container because it's like if it's mixed with, oh, Danielle has it. So it looks like, kind of looks like a little flower with leaves, not like something that would make you think like, oh, that's irradiated, but again, there's still a lot of opponents to it. Irradiating vegetables, they say, like makes them more mushy, kind of damages them a little bit. So they think that it doesn't work well for fruits and vegetables. The meats I told you that, like for chicken and stuff, they said it made it smell like burnt hair. But right now, I guess there's a couple of states that have a couple of grocery stores that sell irradiated meat. That is as much as I could get out. For sure, they do irradiate spices. And if it's mixed with anything else, they don't have to put that sign on it. So if like they make your food with irradiated spices, it doesn't have to have that symbol on it. It's only if the whole thing has been irradiated. Obviously, they're trying to do that because people in general and the public are just not really comfortable with the idea. There are studies that have shown that mice that, ate all this irradiated food developed, cancers and stuff. But then there's as much, research on the other side that doesn't show that kind of thing, but it just still keeps people wondering if there's just something that we're not really aware of that the radiation can do. Also, there's an organization, it's like Food Watch, Food and Water Watch, Water and Food Watch, something like that. It's like kind of a well-known organization that does a lot of lobbying and stuff like that. They said also that the use of radiation on foods increased the growth of certain organisms and also killed off like good organisms, and they had a lot of other reasons why. Also, when I was just happened to be looking it up, I came across like a triclosan thing. Has anybody ever heard about that? That's the one that's in hand sanitizers, because they're trying to get that banned because it's also like that. The biphenyls, that are supposed to be anti-hormonal. So supposedly triclosan also is like that too. So it's supposed to be something that really is unhealthy for that reason too. But remember, I was mentioning that they're trying to have like the second generation come out, which is based on, does anybody remember? Based on what chemical? What?
The benzene. Is that wrong?
Bactine.
Yep, benzalkonium chloride.
Ha!
Right, and so that might be part, I didn't know anything about this triclosan thing. I just knew that it was also leading to the development of some organisms, so that was kind of interesting today. So I didn't really find out too much more about the radiation. It's been going on for like 30 years though, where it's been approved, they think it's fine, but really it's really being used very, very little on anything else but the spices. But it might end up, being used more. And that they said like sometimes you can find like irradiated strawberries and stuff like that, but it's supposed to, at the grocery store, they are supposed to post a sign. But there was also some other thing going on too that said that they could call something pasteurized, but it could actually really be irradiated. So again, it's just like mixing words, because like we're all fine with pasteurizing, and we are all kind of like don't know what to think about irradiation. So it'd be like, oh well, they could call it pasteurized because really it did the same thing as pasteurization, killed off pathogens, but not necessarily killed off everything. We'd be happier, right? That's why like you have to like keep reading all the time. It could make you crazy. Bacteriocins can be used also. So different types of bacteriocins can be added to foods to help prevent other organisms from growing. Nisin was one of those examples that comes from Lactococcus lactis. So you see here as an example used in low acid foods to inactivate Clostridium botulinum. Bacteriophages. So more and more research in that area. So all those last slides were all just related to different things that can be used to help prevent growth. And the same thing with packaging. And that takes us to foodborne illnesses. So I think I can say kind of what I need to say since we talked about lab stuff already. But as far as this number, I think that I've thrown this number out before, 76 million cases per year of foodborne illnesses. That's an estimate because definitely they don't all know these types of pathogens. And what I really want you to be aware of is the difference between foodborne infections and foodborne intoxications. And you might wonder why it would be important to know the difference between these. One of the things that I've mentioned before has to do with the fact that if something is an infection, it usually takes a longer onset time. So you usually have to start looking at what did somebody eat 2 days ago, or at the very least, at least 24 hours ago, not what did they just happen to eat. Knowing whether something is an intoxication or an infection is important all the way around. It's important for preventing additional illnesses. So like in an investigation, you might be really trying to prevent other people from eating that food and getting sick. So you want to know where to go. Like, do you start like looking at what somebody just ate, or do you start looking at what somebody ate yesterday and the day before and the day before that? So that's one thing. The treatment is going to be different. If something is an infection, it's usually going to be more lower intestinal type symptoms, and it means an organism is growing. And while that organism is growing, it's producing things that are going to make somebody sick. Sometimes it's producing toxins too, which is still different because that's just still part of the organism. So in an infection, the organism is making somebody sick. So you're going to be more likely to treat somebody with a foodborne infection with something like an antibiotic to kill off the organism. If they have an intoxication, they've gotten sick from eating a toxin produced by the organism, so using an antibiotic isn't going to help them. So understanding the difference between these two categories tells you a lot already. And looking at somebody's type of symptoms is going to help from the very beginning too. Because again, an infection takes time for the organism to grow. So you've ingested some kind of food that's contaminated. You get that organism in an infective dose, and it starts growing, in your intestines. So it's made it all the way through to your intestines. You're going to more likely have lower intestinal type symptoms because it's in your intestines, it's not in your stomach. Then the doctor is going to have an idea how to treat you, and the public health people are going to start knowing exactly where to look, or at least it's going to narrow their range. Yes, Michelle?
You said antibiotics treat infections, but don't treat intoxications. So what treats intoxication?
It depends, like usually with an intoxication, they're going to treat you with, it depends, like if it's something like a neurotoxin versus like an intestinal type of toxin like staph. They might just treat you with fluids or something. If it was a neurotoxin like botulism, then they'll give you an antitoxin. There are antitoxins for botulism. They usually are like kind of too late to get, but yeah, Sean. Well, if something like staph, it's not, you've ingested the toxin that's making you sick, not the organism. So it doesn't do any good to kill off the organism because that's not making you sick. The organism produced the toxin in the food and you ate the food. So even if the organisms are still there, it doesn't really matter because they like basically poisoned you by the toxins they produced.
With something like botulism, can you still get an infection from it? I feel like they still would give you stuff to treat possibly.
Not an antibiotic though. For botulism, the only exception for that might be something like infant botulism, because the organism is producing the toxin in the infant's stomach or intestines. But other for regular botulism, no, they wouldn't treat you with an antibiotic because the organism is an anaerobe. It's not going to be like causing a problem for us. If you ingest the organism, botulism, the Clostridium botulinum, it'll like, if it's in its vegetative state, it'll just die off in your stomach acids. Okay, an antibiotic like has to, like you have to have like a growing organism there for it to make a difference. Whether or not, like when you say, oh, I feel like they would still give you one, doctors practically gave people antibiotics for just about anything before. So like we probably have a sense, you know, that for years, like every time you went to a doctor with any kind of anything, they gave you an antibiotic. But that's not really how antibiotics really should be used. So like if they gave you an antibiotic for something like that, they would be giving it to you for no reason at all. Because it's like they regularly gave antibiotics for viral infections. When questioned about it, they say it's to prevent development of a secondary infection. Like say for instance you had a cold and because you were so sick, now you got some other kind of infection just because your resistance was lower. That's not really a good reason. Because actually when you kill off your good bacteria, they're part of what keep you healthy. So it could actually make you even more likely to pick up something. You know, it's just taking us a lot of years for the medical profession to get comfortable with that. Because if you're a doctor and somebody comes in to see you, you feel more likely you better treat the person for everything because you don't want to like not treat them. But other than that, like, no, you wouldn't need one for that because it wouldn't do any good. Okay, so foodborne infections, and you can see like it's coming up here. It's ingestion of the pathogen followed by growth or tissue immunity and or release of toxins. See where it says and or release of toxins? That doesn't make it an intoxication though. When we get to the next part where we're talking about the disease cycle and what makes a virulent organism, sometimes in there causing, you know, problems for you when you're having an infection, some of that comes from toxins that they produce in your body while they're growing. Okay? And in foodborne infection, the organism is growing. Part of the damage that causes you comes from something like the fact that it's invaded tissue and it's producing toxins. Like say for instance, the Shiga toxin. Okay, Shigella is a foodborne infection, but it produces a toxin that causes part of the damage. That doesn't make it an intoxication though. Okay, an intoxication is when the organism produces a toxin in the food and then you ingest that toxin. So for the example, Staphylococcus aureus. Okay, if a food gets contaminated with Staphylococcus aureus and it's left sitting, so it's had some kind of mishandling, it's left sitting for over 4 hours. 4 hours is always the magic number in foodborne infections. Okay, like when you grew up, your parents could make you a sandwich in the morning, you could take it in your lunch, it didn't need to be refrigerated, right? You ate it at lunchtime, everything was fine. But you wouldn't leave it there all day. Of course, I know things are different now, everybody has like an ice pack and stuff like that. It's not like when we grew up and you had just like the brown bag or a lunch pail or something, but it really is okay for food to be out for 4 hours. That's kind of, you know, the magic number. If things get, not at a restaurant, but in your own home, but if something is contaminated with staph and it sits out for longer and it grows that toxin, or it produces that toxin while it's growing, that toxin is heat stable, very heat stable. So that was the one that we were mentioning. You could fry it again, you could bake it, you could boil it, you could do anything. The organism all dies off. The toxin is still there. So you eat the food, you're not even eating the organism. So say for instance, like, oh, you got the pizza, you had like a little party for your kids. It was like they're older, so it's a slumber party, they're like 10. The pizza's sitting out all night. Somebody eats it in the morning, right? Something like that. Or maybe it stayed out all night, so you thought you'd be safe and you'll put it in the oven again, right? Just to kill everything off. If the toxin was there, you could kill off the staph by putting it in the oven. The toxin is there. The toxin is what's making you sick. In botulism, the organism could be killed off. The toxin is there. The toxin is what's making you sick. that's an intoxication. You see the difference? The organism doesn't even have to be alive or around anymore. The toxin was produced in the food. Well, botulism, you can cause you can basically, let's see, I'm trying to think of the word, it's not kill off. You can cause the toxin not to be poisonous anymore by boiling something, boiling whatever it is in there. It's heat labile, it's not heat stable. But the point is that it's still the toxin making you sick, it's not the organism. Right? You understand that part. Because things could be heat stable, or they could be killed off, or they could be, what is the word? Neutralized, yeah. You can neutralize the toxin. Some of them are heat labile, they're sensitive to heat, so you can destroy the toxin with heat. Okay? But if you, for some reason, didn't do that, the point is, one, the organism is making you sick, the other one, the toxin is making you sick. Okay, the toxin that was in the food. For sure, on the exam, I'm going to ask you to explain the difference between foodborne infection, foodborne intoxication. I will ask you to give me a few examples of each. And I'll also ask you why it's important. Because I do want you to understand why I'm making a kind of a big issue about this. Because the types of symptoms you get are associated with whether it's an infection or an intoxication. And these are generalities, because remember there's always exceptions. Okay, how the onset time is really different. The onset time is much longer in an infection than an intoxication, because in an intoxication, you ingest it and there's a poison in it. So it's going to make you sick in a much quicker time. It doesn't have to grow. It just has to get into your system. And the types of symptoms you have are more likely to be upper digestive, or in the case of botulism, it's a neurotoxin. So it causes nervous system problems. What was your question, Sean? You got your hand up. Oh, I'm sorry. I didn't mean Sean. Sorry. Sorry, I'm looking at you two or something.
Would the example be like that?
Say that again.
Would an example like be like what we just talked about?
Oh, for examples, I'll want you to know those organisms that we talked about, like Staphylococcus aureus, botulism, those are two, Bacillus cereus, and then all those other things, like E. coli, Salmonella, Shigella, those are all the infections. So it's important for preventing additional disease, it's important for understanding how to treat it. They're all interrelated. Yeah, that's one of them, the Shiga toxin. That would be an infection. Yeah, and that's an example of the fact that if you have a virulent type of organism that you're infected with, one of the things it does is produce a toxin in your body that causes problems. Like for instance, the thing that you did your paper on, the necrotizing fasciitis, it produces a toxin into the surrounding area that prevents it from healing, that destroys tissue and everything. That's part of its virulence. So I know that kind of has an overlap there, but I think you can understand the difference, right? Because one is the toxin was already produced in the food. The other one just happens to mention that particular infection, that organism can produce a toxin as part of how it causes destruction to you. Listeria was another one. Here's the one with intoxication. Clostridium perfringens actually can kind of do both. Bacillus cereus. These are all ones that we talked about before. There's other examples in here. There's algal toxins like the paralytic shellfish poisoning that comes from an algae, aflatoxin and such, which I'm not going to ask you about on the phone. Okay, but I thought you'd just be aware of that. Okay, so that covered food and water. I did want to show you this before we go to lab, just because I looked it up.
That's not really something I thought earlier. You're crazy. Maybe that's what you laughed
Yeah, you did.
Maybe it's your calculator. Hmm? What do I have? Give me the possible punch.
Here?
Yeah.
Just because I was looking around for the stuff on radiation and such, just going on the FDA's website. And they had like the, you know, for food, this is under food, recent outbreaks. And because this goes along with the things that I've said before. So this is like, and this is from May 8th, just this year. And it's talking about an multi-state outbreak of Salmonella. Salmonella St. Paul, that's just the particular strain linked to cucumbers. Okay, so when you go through and you read here, it says the multi-state, blah, blah, blah. A total of 81 people infected with the outbreak strain of Salmonella St. Paul have been reported in 18 states: Arizona, Colorado, California, Idaho, Illinois, Louisiana, Massachusetts, Maryland, Minnesota, North Carolina, New Mexico, Nevada, Ohio, Oregon, South Dakota, Texas, Virginia, Wisconsin. Nationwide, 16 people have been hospitalized, no deaths reported. Okay, so when they go through and they give a little bit more information. This came from one place. This is what I'm talking about when I say things are different. This is a perfect example of that. 18 people, 18 states, that's just what's been reported. Because somebody can get salmonella and never go to the hospital, never even go to the doctor, just recover on their own. Some people could go to the doctor, but it's never tested because they go, they get an antibiotic. By the time they have a stool specimen, the antibiotic has killed off a lot of it. They don't get results. So these are just the ones that they really spend more time investigating at the hospital. It gets reported, it gets investigated. 18 people, 18 states, one place distributing cucumbers. Okay, not the same as before when things were grown locally and somebody got them. Do you see what I mean? Okay, and it just so happened to be that they showed two different ones. The other one is E. coli 0121, which is just another strain of the STEC. This is a Shiga toxin producing one. Of this one, okay, it's where is it? Rich Products Corporation of Buffalo, New York, has expanded its earlier recall of certain farm-rich and market day products, including all products produced at its Waycross, Georgia plant. Okay, so this is like one company, one plant. It's a multi-state E. coli 0121 illnesses, possibly linked to those farm-rich and market day. 35 persons infected with the outbreak strain of Shiga toxin producing E. coli 0121, STEC 0121, have been reported from 19 states. And I don't think you want me to read all the 19 states alphabetically. one facility, one company, 19 states reported. And like have you even heard about this on the news?
Nope.
Right, okay, so this has been going on, this was posted May 10th, just recently. It's already been going on because they just increased this, it started April 5th. You guys haven't heard anything about this, right? This kind of stuff is always happening. You think that you always hear about all this stuff on the news, but you don't really hear about it. It just depends on like what happens to get the attention. And you'll get this like on our website and such, but still like, I'm not saying that the news doesn't report it at all. It's just that, okay, so it was mentioned on the news. It's not something that the general public is aware of. If you go on here every day, you see things, like we could go to see all these different things about what things in the past 60 days have, like here, where's the recall? And then you could just start looking back, okay, so 5, 10, some of these things are things like undeclared things, undeclared eggs, but here's salmonella. And here's more undeclared. Wait, how did I get on that one? Oh, because I'm on all.
Where?
Okay.
Brandy, Whole
Here's
Foods.
salmonella for that one. Here's Listeria, 430, Prime Foods, salmonella for tahini sesame, salmonella for bird food, Listeria for herring and salmon. And this is all like in the past 60 days. You know, there are other things too. So you can go through, here's that one, Farm Rich, the people one, go up to. And there it is again up there. You can go on and just read this regularly. There's all kinds of things that are always happening, but the point in this is that when it does happen today, it's something that affects a lot more people. That one plant makes food that gets distributed all over the United States. It's not like the local food producer that made things for your town, for your city. And that's why when you have these discussions of, oh, we always used to do this. When you have a company that's a small company making just for local things from local things, you didn't have the same concern. It wasn't going to be something that was going to affect that many states, that many people. And when it does affect a lot of states, it's not that easy to get all that information. So it just kind of, I just kind of came across that and wanted you guys to have an opportunity just to see that. And again, I know that they cover things on the news, and like I said, I get these alerts every day because I do